used to summarize their academic careers, the prolific academic careers, and give an introduction, but I'll still try. So, Professor Vinod Kumar has had a prolific academic career which does not need any mention. Sir did his PhD in 1981 from Banaras Hindu University and then went on to give his academic contribution to several institutions and universities. To name a few, he has been the visiting fellow of Max Planck, Germany. He has been the visiting professor, Department of Biology, Texas University. Sir has been a recipient of several prestigious awards. Very early on in his career, he received the Young Scientist Award from the Indian Science Congress Association in 1986. He was the Young Investigator Award from the Gordon Research Conference in Pioneer Cell Biology in 2000. He received the P. Govindarajulu Gold Medal, Society for Reproductive Biology and Comparative Endocrinology in 2006. Vijay Usha Shore Scientific Research Prize, University of Lucknow in 2008. The Platinum Jubilee Lecture Award from Indian Science Congress Association 2010. Very recently, Sir has been the recipient of the prestigious Ascof's Ruler Prize in 2022, and currently he is the UGC BSR Fellow in the Department of Zoology, University of Delhi. I welcome you, sir. Our next chair is Professor Shivnath Majumdar, currently Professor, Dean and Proctor, Faculty of Life Science and Biotechnology, South Asian University. Sir did his PhD in 1997 from the Postgraduate Institute of Medical Education and Research, Chandigarh. Sir has been the recipient of INSA Teachers Award. He is a member of several prestigious societies: Indian Immunology Society, Indian Society of Cell Biology, Indian Society for Molecular Immunology Forum India, to name a few. He has given his academic contributions to Vishnu Bharati, to Banastali Vidyapeeth, as well as to our very own Department of Zoology, University of Delhi. Sir has mentored several students and has several research articles to his credit. I welcome you, sir. And with this, I would like both of you to take on the section. Thank you.
Sal in Google, and I am retired now, so fused bulb. So that doesn't matter, but I'm really privileged to co-chair this session with my senior colleague, Professor Shivnath Majumdar, and I proudly hand over this mic to him because he is the right person for this particular session. So here on Professor Shivnath Majumdar will take care, and I'll try to listen, catch up some of the immunological. Thank you so very much.
Thank you. Good afternoon. Namaskar. At the outset, I would like to thank the organizers for giving me a chance to participate in this session, and I hope we'll have a nice time together. Our first speaker is Professor KL12 Srivastava, and it makes me it's very difficult for me to introduce Professor KL12 Srivastava because Anju and me, we were friends. We used to fight together, we used to do everything together. We had a wonderful time together in this department. So Anju, she is now the professor in the Department of Zoology, and you have everything over here. You can read it out. Thank you.
It's really, I'm very happy in fact that I'm getting the opportunity to speak in front of my teachers. There are three teachers whom I have actually learned a lot: Professor Bharat Agarwal, Professor Madan Mohan Chaturvedi, and in fact coming in research is because of Professor Madan Mohan Chaturvedi, because the interest in science actually was inculcated by him, and not because he's sitting in front I'm saying that is the truth, sir. I've always been inspired by your way of teaching and the way you have inspired the students, right? When we were also students, and all the students here would acknowledge. And as far as getting into the field in which I am now working is the contribution of Professor Bharat Agarwal, because I had the opportunity, good fortune to be in his lab for a couple of years, and that is when I got interested in immunology and inflammation, and that is from there only I actually picked up this area. Now the work which I'm going to present today is a newfound interest. Earlier I used to work with macrophage. My PhD supervisor, Professor Ajit Sodhi, and I shouldn't forget to actually mention him and to actually pay my respect to him, because he is another person who has been very, very instrumental in where I am at this point in time. So the newfound interest which I say is neutrophil. Up till now we have been paying lots of attention to macrophage, and macrophage particularly because they have the tendency to infiltrate in the areas where inflammation actually is. Whatever kind of inflammation, we will find macrophages sitting there. So, and especially in tumor, there is infiltration of these macrophages, and I was very much interested, and I used to work, and I'm still working with macrophages. But during the course of studying macrophages, there were few observations which actually attracted me towards neutrophil. And when I started looking, and this is where I would say that these are some of the limitations of the way we work. When we work, we become so focused that we do not look beyond what we are working. So I used to get focused, my lab used to be focused only on macrophages, and we were not looking on the other cells. But during the course of my journey, I got to see that when the cancer actually develops, there is tremendous change in the number of neutrophils, which we actually found out not because we were looking for neutrophil, but accidentally. So here, and then when we started looking into the literature and other things, what we and during our research also, what we find that when we say inflammation, what we are actually talking about the fire within, and as long as this fire is in control, it is good, it helps us. But the moment it becomes uncontrolled, it can lead to several problems. And so this is what it says. As long as it is acute, it helps us. The moment it becomes persistent and chronic, it can actually result into a number of different kinds of diseases. And the other key player in inflammation, all of us know, that is neutrophil. And when you look into inflammation, you will find right from head to toe, any kind of inflammation in any organ, you will find inflammation is resulting into various kinds of diseases impacting various organs. So, inflammation, as long as it is acute, it can actually bring about a beneficial effect. Once it becomes chronic, it starts causing problem, and then later on it can actually become systemic chronic inflammation, and that might actually result into various kinds of issues. It may have a systemic effect, and as you can see in this case, as you can see, if we talk about take the example of visceral adipose tissue inflammation, this inflammation may cross-talk through the adipokines and other effector molecules to the brain, and through the brain various organs are getting impacted, and these organs in turn may cross-talk with the inflamed tissue, and it might actually result into various spread of the inflammatory condition. Various organs may get impacted, and the major role played may be via the phagocytes, it may be macrophages, or it can be neutrophils as well. And as this cartoon shows, that there could be number of mediators which are released, but this crosstalk is majorly mediated by the neutrophil, and a very beautiful review article was published recently in 2020, which actually shows what are the various mediators, and especially the neutrophils play a very important role in the crosstalk. So, what impacts, what are the causes? And all of us by now, through the course of this whole discussion, have come to now realize, and we already know, that be it xenobiotics, disturbed sleep, chronic stress, diet, dysbiosis, obesity, physical inactivity, or chronic infection, you name all these lifestyle related things may actually impact these cells, and later on it might actually impact all our organs. So inflammation does have a systemic effect. And the key players are your myeloid cells, which originate in the bone marrow, and from bone marrow various cells are originating, but today we will confine our discussion to the neutrophil. Now, as the beauty of the cells of the immune system is that they show tremendous plasticity. And this plasticity basically means, what I mean here is phenotypic plasticity. That means depending upon the area where they are living, or where they are going, or from where it is coming, it may show different phenotype. So so like any other immune cells, even the neutrophils are found to show various phenotypes, say naive neutrophil, or you say T cell suppressive neutrophil, or neutrophil which may show enhanced netosis. So, and all these different phenotypes can be identified depending upon various surface markers which they express. And now people have started realizing the importance of these neutrophils in various disease conditions. All of us know they originate in bone marrow. Through successive course of development, they actually develop into neutrophil. There are around 50 to 70% of the cell population are the neutrophils, and what is very interesting is only 1 to 2% of the mature neutrophil come into circulation, and they remain in the circulation whenever there is requirement, whenever there is a stress condition, there is a call, then only these neutrophils infiltrate into the tumor, into the inflamed tissue, and may bring about resolution of the problem. They may function via, they have the important point of neutrophil is that they have lots of granules or the cargoes which can bring about their effector function. They may function via degranulation through phagocytosis, and newfound technique is that they may show netosis, wherein they can send their cargo and specifically through netosis to trap the various pathogen and resolve the problem. So neutrophil plays an important role both in health and diseases, and when at the site of infection, once the neutrophil function is over, it should be cleared, and so as to maintain the tissue homeostasis. But as long as they remain there for prolonged time, they accumulate, they may cause actually tissue damage, and that can actually initiate chronic inflammatory state. So, and people have found that if there is elevated or increased recruitment of neutrophil or heightened immune function of these neutrophil, excessive release of the granules which they actually have, you will find that they are being implicated in multiple diseases involving various organs. So this is just an example. If we talk about respiratory disease, neutrophil have been recently in COVID-19, it has been implicated very severely, and then you can see they function via the release of the cargoes, the reactive oxygen intermediates, proteases, and other things. So now the question is whether neutrophil plays any role in cancer, and why this question was that until very recently it was said that they are neutral, they have no role to play as far as cancer is concerned. But recently people started paying attention, and the major attention was paid into tumor infiltrating neutrophils. So the ones which are infiltrating, they are activated and they may actually cause certain kinds of problem. Alright, but not much was actually known, and not much has been worked on the systemic effect of neutrophil. And we actually wanted to study that whether these neutrophils which are there in an individual, especially we use mouse model, whether the neutrophils which are found there, do they have impact on the tumor macro environment? And we wanted to actually study that. The question was whether and how neutrophil contribute in mediating any kind of systemic effect in the cancer. Alright? So we tried to look into the numbers and whether they are infiltrating into the into various organs or not, and if they are infiltrating, what is its function, what kind of phenotypic variation they are showing. So in order to assess its function, we actually utilize a mouse model wherein we actually studied the neutrophil on various time point of cancer growth, that was on day 0, starting point 7 and 14, so as to see with progression what changes are actually happening. And this whole study had started with this observation. The story which I said, it came later to this part. Why? Because one of my PhD students, when she started, we actually started looking for macrophages, but when I asked her to look into the blood profile, what she found that when you take a look into the blood profile of a tumor growing mice, you are seeing that in the tumor, in the blood, there is heightened number, a very high increase in the neutrophil count. And as the morphology shows, the neutrophil number was very, very high. And then we looked into the TLC, and then we also did the DLC, and we find that the polymorph numbers are actually, or the neutrophil numbers are actually increasing gradually with the increase in the with the progression of the cancer. On the contrary, the lymphocyte number was actually going down. And when we looked into the literature, we found that this polymorph to lymphocyte ratio actually is a predictive marker to say whether in cases of growing cancers. Just give me 1 second. So then whether or not it is a neutrophil, we actually checked for the marker, and Ly6G was used, and as you can see, with progression 7, 14 and 21 days, it is increasing in number. We did FACS analysis, that also showed that the neutrophil numbers are increasing in the blood. Then the next thing which we could have done is to see the enzyme function and of the various such as liver function, kidney function, and what we saw that with progression, the enzymes were also showing variation, especially the ALT, the amount is getting high. Similarly, the urea is showing increase, albumin is showing increase. So there were indications that the organs are actually getting. This was the preliminary data. Based on that, we actually did the whole story. Then the question was that do we see these cells or any kind of but if your organs are showing different function, there is increase in the enzyme level or there is difference, we looked into the histology of it, and what we found to our surprise that in all these organs, lungs, liver, or say for example spleen, or you talk about peritoneum where we had actually injected the tumor, in all the cases we are seeing there is increased infiltration of these polymorphs. Now the question was that whether these polymorphs are neutrophil. Similarly, we did the Ly6G staining for these, and we found there was tremendous increase. If you see here, look at the peritoneum, look at the lung tissue. If you look into the kidney tissue, again you can see that there is very heightened increase of neutrophil infiltration. There was significant increase in the neutrophil infiltration. So in this first observation, the preliminary data which we were actually, which we saw, the observation was that there was increased count of neutrophil in the peripheral blood. There was tremendous increase in infiltration of neutrophil in the vital organs. We were not looking into the tumor infiltration, we were looking into the organs, the peripheral organs, the places other than the tumor. There was disturbed histoarchitecture and altered biochemical enzymes. So the question was to examine whether systemic impact of neutrophil, what is because of the, is it because of the systemic impact of the neutrophil? And then the most obvious thing was to find out what is its functional change. Is it showing heightened activation, or is it contributing because of the neutrophil function? And as all of us know, neutrophil actually have, upon activation, they have azidophilic granules. You have another granule such as collagenase or gelatinase or there are number of enzymes which are actually they're present in these granules, and upon activation they may work via granulation, phagocytosis or netosis, so in order to bring about the killing. So this is how it works. So again, in the same setup, we try to look different enzymes so as to understand the tissue change which is happening. Is it because of the release of these enzymes in these tissues? So the neutrophil elastase is the most abundant enzyme present in the acidophilic granules, and these hydrolyzes the elastin and other extracellular matrix components such as cadherins, fibronectins and all. So we wanted to look into that, and if you look into this, if you look into the gradual progression, you can see how much increase is there of neutrophil elastase. Similarly, we looked for MPO, another important enzyme, and you can see exactly similar trend that there is increase in the MPO with progression of cancer. And if you look into this is the nucleus, or this is the blood cells of the neutrophil obtained at different time points, and the most remarkable thing which you can see here, that the nucleus is actually getting highly fragmented, and this is the hallmark of the change in the phenotype of the neutrophil in cases where the cancer actually is progression. So even like macrophages, the neutrophils are actually N1 or N2 type, which is one is known as tumor promoting, the other one is known as tumor supporting type. And if you look here, if we actually looked into the Ly6G staining is for confirming neutrophil, MPO and NE staining, and when you look into this, you can see these neutrophils are expressing, the ones which were present in the peripheral blood are also expressing these enzymes in much higher quantity as compared, and the phenotype is also getting changed. So, if the infiltration contributes to high elastase with progression, all right. In the blood, in the ascites where the tumor is growing, as well as in various organs, liver, lungs, spleen and peritoneum. We also looked at the transcriptional level, and as you can see, DL actually says it is the Dalton's lymphoma which we have utilized, and you can see how much increase in the expression of messenger RNA level is there in the cases of tumor. Now, similarly, if you look into the immunohistochemical staining of the tissues, you can see the same situation in the liver, in the peritoneum. The upper panel is for control, and the lower panel is for the lower panel is for the tumors. And if I go beyond spleen and lungs, you can see in the tumor condition how much increase in the enzyme levels are. Similarly, MPO, you can see at the transcriptional level there is increase in the expression of MPO, and you can see through immunohistochemical staining as well that there is increase in MPO expression in all these tissues. So, and then this is another very interesting point, the ROS level in these organs, liver, peritoneum, spleen and lungs. You can see how much increase in the ROS level is actually seen in the cases with increase in tumor development. Then comes whether the collaginases and gelatinases are also showing any change. The gene expression levels can also shows the similar trend, whether it is MMP 8 or 9, it is showing increase. We did the gelatinase assay in order to confirm the activity. There is increase in the activity in the tumor tissue. So with this, we were very convinced that the neutrophil infiltration contributes to high elastase release in the vital organs, MPO release, as well as there is tremendous increase in the ROS levels, and there is increased activity of MMP 8 and 9, which actually suggests that the changes in the organ function probably is because of the infiltrating neutrophil, and it might be contributing to the damage. And this part of the story very recently got published into the immunology letters. Now the question is, and this is a very, very basic cellular study, no molecular study I have done here, no signaling aspect. This is just to see how exactly the neutrophils may be playing a role in actually having a systemic effect. So this, we were kind of convinced. Now the question was whether this whole, because these enzymes are also produced by the macrophages. So what is the contribution of macrophage? We are showing these enzymes in the enzymes. So is it being contributed by the macrophages or by the neutrophil? But what we are seeing, and we think and we assume it needs to be actually ascertained, but what we see, because there is a correlation, there is very heightened increase in the neutrophil count, so probably these are the ones which might be contributing, but this is worthwhile to find out that whether it is being contributed by macrophages or not. Next, we wanted to see whether this can be actually tamed. These are hyperactive neutrophil, and can it be tamed? And can any immunomodulator actually bring about some sort of calming down of these neutrophil, and can it be useful in actually the in survival, or maybe it may be helpful. So we actually utilized Tinospora cardifolia, and this we used a crude extract, methanolic extract. We did not do anything except for we used the extract. And in the same setup, we actually used this extract at different time points. We injected the extract right at the time when tumor was being transplanted, so as to see its preventive effect. And in the second set or the group, we utilized or gave the mice after 7 days of tumor growth, and then later on after 14 days of tumor growth. So just to see at from what till what level the Tinospora we are using may actually have an effect. So if you see this graphs, one has to pay attention is to the is the way that this 0 represents simultaneous tumor transplantation and TCE administration. This is after 7 days of tumor transplantation, and this is after 14 days of tumor transplantation. And these are tumor mice. All the parameters of the tumor mice is represented by DL, right? And then when you see, if you look at the 0 time, when you are in any parameters, you will see that these when it is given along with the tumor cell, when the tumor was being transplanted, Tinospora was found to be highly effective in preventing the tumor growth. We will see one by one the various parameters. So this is the TLC count, the total leukocyte count. These are the polymorphs, and these are the lymphocytes which was found to be significantly reduced, was much better when it was actually, so the blood profile was seen to be actually reclaimed or restored, right? Similarly, if you look into this is the DL condition or the tumor condition. If you give simultaneously, it is more or less like the control. After 7 days of tumor, when the tumor has grown slightly, still it has an effect, but after 14 days, you don't see that kind of an effect. Now in this case, this is the 0 day, 7 day and 14 day. Look at the 0 day, almost like the control. It showed tremendous effect on in preventing, and at early stage also it was found to be fairly effective. If you look into the ascites and the serum for the elastase, again you can see how significantly there is prevention, and even at the early stage it is significantly showing restoration of or reduced release of elastase in the lungs, liver, spleen and peritoneum. Everywhere we are seeing that there is significant protection as far as Tinospora administration was concerned. Which it is again beautifully shown when we looked into various tissues. You can see at the 0, 7-day and 14-day condition, right? These are different. We also did ELISA in different ways. We checked into it. MPO, and this is the most interesting one. If you see the real condition, the reactive oxygen state, and look at this at zero time administration when it is given simultaneously, how significantly actually it is blocking the ROS production. Even at after 7 days of tumor transplantation, the effect on the ROS production is very much actually reduced. So, and then when you look into the MMPs, it regulates the MMP expression in various organs, be it liver, be it lungs, be it spleen or peritoneum. In all the cases, the MMPs are expression is again showing the similar trend. So TC restores the tissue histoarchitecture. This is the normal tissue, and if you look, if you are giving it at the 0 simultaneously, it is able to actually prevent the lungs histoarchitecture, the liver histoarchitecture. Even at 7-day treatment, you can see the effect is pretty significant. This is the normal peritoneum. You see here at 0 time points, you can see in the spleen as well, the normal architecture is very much intact. Similarly, you can see the serum enzyme level. If this is important, the AST almost like normal, urea almost like the normal condition. So the point which I wanted to emphasize, that the TCE, when it is given, and the dose which we actually tried, was able to prevent the tumor progression or tumor growth when it was given together, and when it was given even at the early stage, it had significant impact on the survival of the animal. So if you can, the 0-day, 7-day and 14-day, you can see normally this mice grows somewhere around 24 to 25 days, but the moment you give at 0-day, it is surviving almost like 80 days. After 7 days, if you give, it is somewhere more than 60 days, and even after 14 days, there is increase in the survival. So maybe how it can be given, but one point which is very, very important and to understand, or what we realize, that if we actually have some immunomodulators which can actually have the ability to tame its activity, neutrophil function, it might be actually useful. This is a very preliminary small study just to say that if there are certain immunomodulators which can actually have an impact on neutrophil function, if they are calmed down, they are tamed, then it can have an impact. So Tinospora cardifolia in a preliminary study actually shows that it regulates the neutrophil number in the blood as well as infiltration in the vital organs. It regulates its hyperactivation. It can, and if it is regulated, the activation is regulated, it may restore the tissue histoarchitecture and may limit the growth or the increase the systemic deterioration may be actually controlled, and that might actually add to the survival, better life which a tumor patient actually faces. Major problem actually happens not because of the tumor but because of the deteriorating systemic condition. So if that also can be controlled, then it might be of use for a better survival. So neutrophil counting, so taken together, we strongly support the hypothesis that neutrophil might be majorly responsible for the systemic manifestation observed in the cancer patient, which we actually come from the first part of this study, wherein we are seeing the systemic damage is majorly contributed by the neutrophil, and taming these hyperactive neutrophil might have certain preventive and maybe with combination it might also help in therapeutic approach and may increase the survival of the person or patient and may actually help in better life quality. What could be the future direction? Earlier it was known that there are pro-tumoral role of neutrophil. The number increases in blood tumor site, that they are involved in various parameters of the tumor growth in the microenvironment. Our observations suggest that it has a systemic role to play. The systemic damage during cancer growth is probably because of this heightened neutrophil, and what we need to do is to look into how exactly it can be tamed. One such cautious or thing which we have tried is to look into immunomodulator, can it actually tame it down, and there could be certain others. We need to also look into the fact that from where these neutrophils are coming. Are these neutrophils, just 1 minute, are these neutrophils coming from the bone marrow or they are coming through the tumor? What is the source, what is the root, where exactly and when is it getting activated? Those are the questions which needs to be actually addressed. But one important point is that they do play a very, very important role. So what I always say, that what is the most important thing which one needs to do is to stimulate the phagocyte, be it macrophage or neutrophil. These are the only genuine things. Drugs are usually delusion, and this is my team. Our lab actually works in immunomodulation and tumor development. Major work is on macrophages. This is the girl Kavita who recently got her PhD on this part of the from this part of the study, and others are working on macrophages and other things. There is another area where we are actually diversifying is to look into neurodegenerative diseases, wherein I'm collaborating with one of my colleague in the department, Professor Namita Agarwal, and we are looking into how macrophages play an important role in neurodegeneration using the fruit fly model. These are my students, these are collaborators, acknowledgement for the microscopy facility and the funding, and that's how I pay my gratitude and acknowledge and thank you for listening to me.
We have 5 minutes. Any questions? Yeah, sure.
It's very interesting. Now, is it all simply because they increase in the number of neutrophils? Or if you take individual neutrophils, there will be differential expressions of, you have shown the differentiation of neutrophils. So which stage is the one which has been many studies, and which stage will be more crucial, or there is just simply number per se which is causing all.
So, sir, in generally, that is what I don't understand, but what is very important, that in generally when there is inflammation, especially acute inflammation, when the neutrophil, it is, the tendency of the neutrophil is to reach to the inflamed site. So, these neutrophil, when they reach to the inflamed site, they get activated by the milieu, the environment there, but the moment that they perform their function, they actually undergo apoptosis. They have a very short half-life after activation. But what we have seen in the cases of tumor growth, that these neutrophils, instead of undergoing apoptosis, they are actually surviving and they are remaining in the system for a much longer duration. They don't show apoptosis. So there is not that kind of a case. Now the question is that, is it that more granulopoiesis is happening, and the moment there is more granulopoiesis, more and more neutrophil is being released because there is a stress condition whereby the tumor is growing. Now, are they activated right at that point and reaching to the, because they are infiltrating, they might go into the various organs, and so, or they are going through the tumor. That part of the story I do not know. Maybe we need to actually look into that. I answered.
Yeah. Partly.
So it was, the first question was between Dronacharya and Arjun. I don't know who is going to follow next.
So,
Ma'am, what is the dose basically that you given? And what's the root of Tinosvara? Like Deloitte.
Deloitte. See, in this case, we have injected it, right? But one can also, I also tried giving orally. There, the dose was not actually getting adjusted. It was not constant. Kabhi zyada ho raha tha, kabhi kam ho raha tha. So it was not giving me. But here in this case, I have injected it. Yes, sir.
Anju, I mean, for me, it's just kind of learning. What I couldn't understand that you had, put the tumor and looked into neutrophil, right? Now, is it the neutrophil gets haywire and then tumor develops, or the tumor has developed and it has systemic effect through neutrophils?
Yes.
So, what is your hypothesis?
See, the thing is, what I am hypothesizing at this point in time is that the tumor grows and the neutrophils, because of the stress condition, neutrophils are getting hyperactivated, and from tumor there are a number of mediators, because tumor is actually infiltrated by immune cells, be it macrophages, neutrophil, T cells and other cells.
Yeah, I'm just cutting you because time is not there. But then it can be happening by any stressor.
It can happen,
right?
So that is
then if that's true, then it's not cancer specific.
Quite possible. That needs to be looked. But then the kind of systemic impact in growing cancer condition is much higher.
Bigger enemy is there, damage would be more. So,
yes.
so
yes,
what,
yes, I agree.
I mean, is there a way that you can temper the neutrophil and then your rat or mice, whatever, becomes more susceptible to A versus B disease?
Yes.
That
can be looked at.
Yeah, so that would be there.
Professor Nimita.
Yes, ma'am.
Nice talk, Anju. Just curious, what happens when you administer Tinospora in terms of neutrophil infiltration? Does it get reduced?
It goes down.
It goes down.
It goes down.
Any other question? Next speaker is.
Thank you.
Our next speaker is.
Now request. Thanks.